Morning everyone. This is JIAS Academy Ramburam Chennai. We are giving online and offline courses. Helpline is given here. This is CSIR NET Life Science Unit 8 Part 2. This class is about gene mapping methods. Let us see what is gene mapping method. Lineage maps, tetrad analysis, mapping with molecular markers, mapping by using somatic cell hybrids, development of mapping population in plants. This is what we are going to see in this video. So let us see one by one. First one is gene mapping definition. Gene mapping refers to the techniques used to identify a gene's location and distance between genes. That is what called gene mapping. What is gene mapping? In the sense to use to identify a gene's location and the distance between two genes. The distance between various sites inside a gene also be described through gene mapping. That is, the distance between various sites inside a gene can also be described through the gene mapping. Placing several molecular markers at specific location on the genome is the fundamental elements of all genome mapping. That is, placing several molecular markers at specific locations on the genome is the fundamental element of all genome mapping. There are many types of molecular markers. So when creating genome maps, genes can be observed as a particular class of genetic markers mapped similarly to the other markers. Types of gene mapping. So, there are two types of gene mapping. One is genetic linkage maps and the next one is physical maps. There are two types, major type, okay, genetic linkage maps and physical maps. Both map consist of genetic markers and gene loci. Physical maps, let us see what is physical map. This involve actual physical distance often measured in number of base pairs. Genetic maps are based on genetic linkage information. Genetic maps are based on genetic linkage information. What is genetic linkage maps? In the sense, this map shows the location, which show the location of each gene on a chromosome. So what is that? In the sense, the genetic linkage maps, the use of genetic linkage map is which shows the location of each gene on a chromosome and their relative distance from one another. That is, distance between the gene as well as gene on a particular chromosome. And initially these maps were created by tracing the inheritance of several features like eye color and hair color. Then next you see the blood, saliva or tissue samples from both affected and unaffected family members are used to begin the genetic map. Saliva is the most often used sample in genetic mapping. This is very, very important. So make a note of this point. Saliva is the most often used sample in genetic mapping, particularly a personal genetic studies. A personal genetic studies, the saliva is most often used sample in genetic mapping. Genetic mapping is made possible by crossing over, a regular biological occurrence during meiosis. You know what is crossing over? So during cell division, the chromosome split into two chromatids. They joined at the center by means of centromere. And another chromosome, if you take this chromosome, is also divided into two chromatids. These two chromatids are joined at the center. This is tetrad. This stage is called as tetrad. because it looks like a four chromatids, but it is the two chromatid they joined at the center. They joined at the center and gives this tetrad appearance. Okay, so when they come closer, there will be a sharing of genes, that is crossing over in the sense if they come closer, this arm joined with this part of the chromosome. See, let us take this one. So the tip of the chromosome goes to that chromosome, and that tip has come to here. Okay, this I will show you clearly. See, if you take this is a chromosome. This already I have seen, I have shown in the last class, but once again I am just explaining this. Okay, this is the chromatid in the sense. So the crossing over takes place. Okay, this is the part of the chromosome A, and let us take this is the part of the chromosome B. Now what happened? This joined with this. Okay, and this part joined with this one. So let us make this clear. So can you see this? So this part is now detached with this chromosome and that part. So when they move, you can see this chromosome part goes to this chromosome, that is the chromatid. Okay, chromatid, and this chromatid part is now here. So the genes which is present here goes to that chromatid, and the genes which is present here is come to this chromatid. This is called as crossing over. So that is what we have seen here. So a regular occurrence during meiosis, that is cell division that produces sperm and the egg, at that time this crossing over occurs. This is very, very important also. That's why the offspring have different characters from the parents. Okay, slightly different from the parents. So this is main because of the crossing over. Next, chromosomes line up in pairs in the center of the cell during the first phase of meiosis, when they often stick to one another and exchange the similar fragments of themselves, also known as crossing over. That is what we have seen. Now, genetic mapping makes it possible to determine which gene is present on each chromosome. So this is very, very important while they exchange their genes. So this gene mapping is very, very useful to study which gene is moved to which chromosome. Okay, so this is very, very important. The gene mapping is very, very important to study the exact location of the particular gene of specific chromosome. Based on the distance between the two genes, mapping can also determine which gene is more likely to undergo recombination. So which one undergo recombination and which is not, and which is present in which loci, which gene is present in which loci and in which chromosome. Everything is possible only studying through the genetic map. And physical mapping. What is we have seen there are two mapping, isn't it? One is genetic linkage maps and another one is physical map. Genetic linkage map we have seen. Now we are going to see the physical mapping. So what is physical mapping in the sense? It is one gene mapping approaches that a high degree of accuracy in determining the sequence of DNA base pairs. A physical map provides the nucleotide members and the precise physical distance between the genetic markers. Okay, and The radiation hybrid mapping, sequence mapping, and cytogenetic mapping are the techniques used to produce the physical map. So what are the techniques used to produce the physical map? In the sense, radiation hybrid mapping, sequence mapping, cytogenetic mapping, these are the techniques. Okay, so this is very, very important question which comes in CSIR NET. Now, physical mapping assembles larger DNA sections using DNA markers and DNA fragments. Okay, so physical mapping assembles larger DNA section using the DNA markers as well as DNA fragments. Okay, various physical mapping methods are also available to study genome of multiple sizes and obtain different degrees of precision. And this physical mapping is a common approach used in genome sequencing to acquire an entire genome sequence and determine whether there is any correlation between the specific DNA sequence and phenotypic feature. And another one important thing, what is that? In the sense, the genetic mapping technique, the recombination events are used in genetic mapping techniques to measure the distance between the genetic markers. Number one, random fragment length polymorphism, or RFLP, which measures the differences in homologous DNA sequences to calculate the distance between two markers. Okay, so this method, which method? Random fragment length polymorphism, or RFLP, which measures the difference between differences in homologous DNA sequences to calculate the distance between the two markers. And currently, if you see, the gene mapping analysis targeting single gene disorder use short tandem repeat polymorphism. Short tandem repeat polymorphism, that is STRP. Okay, STRP, this is very, very important. Tandem in the sense which is located one after another. Okay, or it is you can see here, short tandem repeat polymorphism. You can see here, this is 4 times the repetition takes place. That is the same sequence. They repeatedly present in the gene, present in the DNA, or in the DNA sequence, means it is called a short tandem repeat polymorphism. Here you can count it, number 1, 2, 3, 4. So 4 repetition. Here it is 6 repetition. Number 1, 2, 3, 4, 5, 6. Okay. Here it is again 5. 1, 2, 3, 4, 5 repetitions. Repetition in the sense the same sequence repeated again. Okay. Repeated again. So that is called as a short tandem repeat polymorphism. You can see here, see, this is the DNA bases, that is 1, 1, 2, 3, 4, 5, 6. Okay, 6 times it is repeated. So which one? The DNA bases, that is AGCA. Again it is combined with the AGCA, then again sequenced AGCA, AGCA. That is what I have shown here to you. So it is how many times the AGCA here? In the sense, it is 4 times the AGCA is here, the sequence present here, and here it is 6 times it is here, and here it is 5 times the AGCA is present in the sense. So this is called as repeated sequence which is present in the DNA is called as tandem repeat polymorphism, STRP. Okay, next one you can see, that is one more important point. We will see that SNP. What is SNP? Single nucleotide polymorphism. Okay, single nucleotide polymorphism. Here what we have seen, short tandem repeat. Short tandem repeat is this. See, this is the complete DNA. A part of the DNA is here, and it is the repetition is shown in between this DNA. Okay, so this is very short, so this is called a short tandem repeat polymorphism. Next one is single nucleotide polymorphism, which is used in genome wide association and lineage analysis genetic research. Lineage analysis is studied using the inheritance of characteristics as well as genetic signature like SNPs and microsatellite. So what is microsatellite? What is microsatellite in the sense? Short-term repeat polymorphism. This is also known as microsatellites. A short tandem repeat polymorphism is also known as microsatellite. Okay, and now genome web association. Genome web association. See, all these are very important, so make a note of it. That is GWA is genome wide association. SNP in the sense single nucleotide polymorphism. STRP in the sense short tandem repeat polymorphism. RFLP in the sense random fragment length polymorphism. So keep a note for all those things. So studies which one? GWA studies the connections between the traits and markers like SNPs and microsatellites by treating the population as a single family. So this the method which is used to map the gene function of common disorder. So this mapping is used to map the gene functions of common disorder. So GWA, this method, that is genome wide association method, is used to map the gene function of common disorder. You can see this is the human Y chromosome, the structure of human Y chromosome. This chromosome consists of two arms. This is short arm, this is long arm. Short arm is marked by P and the long arm is marked by Q. Okay, it is joined at the center. At the center, that is, there is a centromere. So they have joined and different genes are marked here and their code is also marked here. So this is human Y chromosome. Okay, human Y chromosome this is. So thank you so much for watching this class. Share this video to your friends, those who are preparing for UGC NET, and our website is given here. You can contact to our office and get the details. Thank you so much.